Greetings, students. Mr. Sanger here. Today I want to introduce you to a lab which is a simulation of Hardy Weinberg equilibrium. And the way this lab works is that we're going to use our class as a sample population in order to simulate the changes in allele frequency that occur as a result of various evolutionary processes. During this lab, we are going to simulate 4 different things: Hardy Weinberg equilibrium. We are going to demonstrate the effect of natural selection. We are going to demonstrate something called heterozygote advantage. And then we're going to simulate something called genetic drift. And then finally, you're going to practice using the Hardy Weinberg equilibrium equation to sort of manipulate some data mathematically. So, I have a few kind of sample problems that we can work through that we're just going to skip over for the screencast, and let me explain how this lab is actually going to be done. So for this lab, every student in the room is going to assume an initial genotype of big A, little a. And so each student is going to acquire 4 different note cards, and you'll notice that those 4 different note cards, 2 of them say, if you look at my little picture on the screen, 2 of them say big A, 2 of them say little a. And these represent the 4 products of meiosis for someone with genotype big A, little a. So everyone in the room starts with this same genotype and these 4 products of meiosis. And then we're going to engage in a basically simulation of random mating, where each individual passes on 1 allele to the next generation. So you're going to need to find a partner. And give me a second. Great news, everybody. I found a partner. Okay, come on over. It's Ms. Rutland, everybody. Hey.
Hi, students.
Okay, so Ms. Rutland and I, we both have assumed the genotype big A, little a. We both have the 4 products of meiosis. And what we're going to do is we're going to put these cards behind our back. We're going to shuffle a little bit. We're going to shuffle. And then we're both going to pull out 1 particular product of meiosis. On the count of 3. 1, 2, 3. Go out. And so basically, this is going to be the identity of 1 offspring in the next generation. So let's suppose, we have to decide who is going to be that offspring. So let's have Ms. Rungland. You're going to be big A, little a for the next generation. Okay. But now we need to do it one more time for me. So we're gonna do the same thing again. So shuffle, and then pull out another one. Isn't she great? Thanks, Ms. Rungland. Anyway, so basically, after that 1 round, Ms. Rungland would then assume the identity of another big A, little a, but I would assume the identity of big A, big A. So basically, in our data, after the first generation, Ms. Rungland would write big A, little a as the result, as the next offspring, whereas I would put big A here. And then what we would have to do is get ready for the next round. So Ms. Rungland would keep going with her 4 cards, big A, little a, but I would have to trade my cards out. Since I'm big A this generation, I would have to trade these cards out, and I would have to get 4 of the big A's. Because if my genotype is big A, then the only products of meiosis that I could produce would be big A. Okay, and then I would find a new partner, and we would repeat that process. And then, the first round, 1 of us would assume the identity of that offspring. The second round, the other of us would assume the identity of that offspring, and we would record it here for generation 2. And so we would go through repeating this process until 5 generations are done, and then that would be the end of our Hardy Weinberg equilibrium simulation. Okay, so the general idea is in this large classroom of a lot of people all moving around, kind of randomly choosing a partner. There's no mutation, there's no natural selection, there's a large population size. Theoretically, we should be maintaining Hardy Weinberg equilibrium. And so the question is, will we, by generation 5, will we be at Hardy Weinberg equilibrium? Okay, so once we do that, then we would repeat this to do 3 other simulations. So the second one is we're going to simulate natural selection. In this case, we assume that the little a genotype is lethal and does not survive. So if Ms. Rung and I were to do this and the offspring turned into a little a, we would basically say, oh, that offspring died, and then we'd repeat the procedure until we got a viable offspring. So we're basically selecting against little a, and then ultimately after a number of generations, we'd figure out, well, how does that affect the frequency of genotypes? Heterozygous advantage. This is where the heterozygote has an advantage over the two homozygotes. So we would basically repeat the procedure, and the result is if we produce a big A, little a, that offspring lives. If we produce a, little a, that offspring dies, and we repeat the procedure to get someone who lives. But if we get big A, we do even odds. We each, on the count of 3, put out a number of fingers, like boom, I put down 3 fingers. It's hard to do this with the camera. Let's suppose Miss Rungland was here and she put down 2 fingers. So we each do a number of fingers between, 1 to 5 fingers, and then we add them up. So basically, we have 5 total fingers. And so if they're even, that offspring lives. If they're odd, it dies. So we have to do it again until we get somebody that lives. Okay, so basically the heterozygote has an advantage because they live no matter what. Little a dies. Big A has a 50-50 chance of living or dying. Okay, so we repeat that one for 10 generations. Finally, we simulate genetic drift. This time we take the class and we divide into smaller groups of about 10 individuals. And we say, let's just have these 10 sort of do their own little simulation. These 10 people over here do their own simulation. These 10 over here do their simulation. And with that really small population size, let's see what happens. So that's basically the simulation. And so then at the end, we have a Google form, and the whole class basically goes through and they record what genotype they got for every generation, for every simulation. It's kind of annoying to do that, but we do it. And then finally, we get some sample data that tells us the frequency of each genotype during all of our simulations. And that sample data will then be posted on the website for analysis. And then that's pretty much it. So hopefully that kind of explains how this all works. Thanks.